Hi everyone, my name is Erdem Varol. I'm a postdoc at Columbia Zuckerman Institute, and I'll be talking about decentralized motion inference and registration of Neuropixels data. So Neuropixels is a recently introduced silicon probe that enables large-scale neural activity recordings from hundreds of neurons. The probe is about 4 millimeters in length, and based on how you insert the probe into the animal's brain, you can capture neural activity from multiple brain regions. You can also synchronously record from multiple probes to capture activity from brain regions that are not next to each other. This is especially important for behavioralists who are interested in studying the crossplay between different brain regions while the animal is executing complex behaviors. But a significant problem in neural activity recording during behavioral tasks is that the animal motion causes microshifts in the positioning of the probe, which then causes a mistracking of neurons over time. And when you mistrack neurons, this reduces the number of neurons whose activity can be accurately characterized over the entire time course. So that effectively reduces the population size that you're studying. So to combat the effect of motion in Neuropixels recordings, we introduce a method that takes 3 steps. First, we featureize the recording to generate a movie whose frames can be used to estimate the motion. Then using these movies, we estimate the motion in each frame using a novel decentralized algorithm. Lastly, after we estimate the motion, we undo this effect of motion in raw recordings by registering the voltage signal to positions that would have been had there not been motion at all. So to give further details on the featurization, here on the left-hand side, you see the Neuropixels recording that's been chunked into 1-second time bits. And the right-hand side, you see we generate these histograms from these recordings, from these 1-second time bits of the recordings that captures the depth and amplitude of spiking events. And the importance of this featurization is that this featurization is invariant to the order by which neurons fire, allowing us to generate a representation of data that represents motion without being confounded by firing patterns. So once we have a video of these histogram-based featurization, the traditional route of doing registration involves taking an average of these frames and then setting this as a template, and then estimating the displacement relative to this template using whatever favorite registration algorithm we choose to employ. But the problem with template-based registration algorithms is that they're prone to underestimating the motion, since a single template might not be accurately represent the spiking activity patterns across very long and heterogeneous recordings. Meaning that, let's say in the beginning of the recording, a subset of neurons are active, but towards the end of the recording, a different subset of neurons are active. There's going to be a severe template mismatch, which is then going to cause the displacement estimates to be underestimated. So to accommodate the heterogeneity of firing patterns across long recordings, we instead treat each time bin as its own template and then cross-register each time frame to each other. This gives us a pairwise displacement matrix that you can see on the right-hand side, which can be decomposed to be a function of global positions using this rank 2 rigidity equation that we use. Using this rigidity equation, we can then move from these pairwise displacement estimates to one final global positioning estimate, as you can see in the lower right-hand side. And one thing that becomes apparent in comparison to template-based registration is that this manner of estimating displacement is more sensitive to small amounts of motion, which can be more visible if we go back and forth between the template-based estimates and the decentralized estimates for the same data set. You can see the subtle slow drift in the beginning of the recording that's the decentralized method picks up that the template-based method does not pick up. And the last step after we estimate motion is to do registration. So once we have motion estimates for each time bin, we can then shift the recording values at that particular time point in the recording with a negative of the estimated motion. And once to execute the shift, then we can simply perform linear interpolation or Gaussian process interpolation. So once we establish this method, now we can also apply this to 3 case studies across different probes and across different animals to show how robust it is to different scenarios. So the first scenario involves applying this to a recording on mice, where the probe was actually connected to a mechanical actuator that simulated a sawtooth pattern of motion. So the benefit of using this data set is that we have a ground truth notion of what the motion should look like. In the middle, you see the pairwise displacement matrix that our method estimates given this data set. And on the right-hand side, in the yellow, you see the global positioning that we can extract from this pairwise displacement matrix. And the satisfying thing about this result is that the motion that we estimate very closely matches the ground truth motion that the actuator was simulating in this data set, which shows that our method was able to accurately estimate the motion. Whereas we also see the template-based registration here in the green line, which is the method used by Kilosort 2.5. And you can see that the motion estimates for the template-based method are slightly different than the decentralized method for this data set. Just to do a sanity check of whether this motion estimate we have gives us actually a registered version of the data, we can look at the raster plot of the spikes extracting this data before registration. And after registration, we can see that the spikes that follow the more or less wavy pattern in terms of the depth by which they were observed have been flattened in positioning after registration, showing that now a particular unit that exhibits spiking events is localized to one fixed position over time after registration. And importantly, after registration, we can track the amplitude of individual neurons and see that the amplitudes have also been stabilized. So in the yellow line here, you see the amplitude of a particular unit that we were able to sort out in this data set over time. And it shows that the amplitude of that unit have been more or less been stabilized for the entire time course of the recording. Whereas in the blue, you see the same amplitude of that same unit had we not done registration at all. And you can see this amplitude is high in the beginning and then slowly wanes towards the end of the recording. And in the middle, the amplitude of this neuron also is highly variable because of the zigzag pattern of motion. In the green, you see the same neuron after it's been registered using the template-based registration that's used in Kilosort 2.5. And you can see that this neuron's amplitude has not been stabilized as well as how it's been stabilized using the decentralized based registration. And at the bottom, you see the same neurons spiking waveforms across time. And in the left, you see the unregistered data and the spike waveforms superimposed on top of one another, whereas the blue lines show the spikes of that unit in the beginning of the recording, while the red ones show the amplitude of that unit towards the end. And you can see that this waveform is highly variable over time, which will create problems for spike sorters down the line. In the middle, you see the spike waveforms, the unit waveforms for that same unit across time after template-based registration, and some of this variability has been removed, although there does seem to be some differences between the amplitudes in the beginning and towards the end of the recording. And the right-hand side, you see the amplitude of the same unit after it's been registered using decentralized registration, and it's very consistent across the entire recording, yielding a very crisp template that we can extract from this. The next case study focused on primate data collected by Churchland lab at Columbia. And in this data set, the motion is more natural. There's no actuator that induced any motion. So the motion is purely based on the behavior that the animal is executing. And we can see in the middle, the motion that we estimate on this data set very closely resembles what we see in the raster plot on the left-hand side. And just to do a sanity check again, using the raster plot before and after registration, here's what it looks like before registration. And after we applied our motion estimates to the data and then regenerated a raster plot, we can see that now the units have been more or less localized to one particular location over time, which will enable further down the line downstream spike sorters to be able to sort out these units with better accuracy. Last case study we try to show that our registration routine is general for other types of probes other than Neuropixels probes. And here we test this on neural nexus probes from Nate Saltel's lab at Columbia. And we show that as long as the probes that have some kind of a channel information to be able to extract the histograms that are used to the registration, we can estimate the motions quite well. So yeah, in general, the method that we propose is an end-to-end method that takes in raw data and actually was able to output the same format data that's been corrected for motion. So this method is very modular, can be used in many different pipelines that can use as a front end. And then, for example, you can do this method, you can apply our registration method and then apply it to spike sorting down the line using your favorite spike sorting method. And the code for our method is available in both MATLAB and Python in this GitHub page. Yeah, lastly, I'd like to thank all my the graduate students and the master students who've been involved in the development and the coding of this method, as well as our collaborators across a couple of different labs who provided the mouse and primate data sets and a lot of insights into how we can improve our method. So I'll end with that. Thanks for your attention.